As always, there's some announcement, and today's announcement is that there is a webinar being hosted tonight by Patrick Barber in the chemistry department about the periodic table. You may know that this year is the 150th anniversary of Mendeleev's organization of the elements into the periodic table. So this is a birthday party by the American Chemistry Society, hosted by Patrick here in this room, 7 o'clock tonight. Okay? So if you're interested in chemistry and the periodic table and its birthday, birth year anyway, tonight. So our goals for today are to finish up the somatosensory system with some review, and then move from the organization of the spinal cord into the organization of the brain, and then discuss for a moment about modulation of somatosensory perception, especially pain, by endogenous opioid neurotransmitters, endorphins and epinephrines. And then we'll start the visual system. So we'll move from the somatosensory system into the visual system, a completely different sensory system, starting with some anatomy, some anatomical physiology related to focusing the eyes and constricting the pupils to limit the amount of light coming into the eye, and then, if there's time, getting into the organization of the retina and the mechanisms of how your retina converts photons into an electrical signal that your brain interprets as vision. Before I go on, let me make a comment about the mid-semester reports that you may have seen. The mid-sem evaluations are available to you on Banner, for not just from me, but from all your courses, so at least everybody who has submitted a mid-semester evaluation. So in my case, the mid-semester evaluation includes a letter grade, which is where I see your your weighted average standing right now, and then a statement about what is included in that letter grade. Problem set 1, quiz 1, quiz 2, exam 1, and the lab so far. And let me make a point that the weight of the lab, which is beneficial to your grade, is a little heavier now than it will be at the end of the semester because of the way, just because of the arithmetic. So the beneficial effect of the lab is greater now by a factor of 2 than it will be at the end of the semester. So that helps your mid-semester grade look better than it would be if, in some cases, not everybody, in some cases it looks better because of the beneficial effect for everybody of lab participation. Okay? So the mid-sem reports just has the basic letter grade, and then if you want to see the breakdown, go to Moodle. The Moodle page has the grade breakdown. Okay, let's move on to today. So you'll recall, this is review, that pain, temperature, touch, pressure, position, information from the body, from the skin and from the muscles, enters the spinal cord through sensory afferents. We'll start to use that word more and more. Afferent means a neuron that is bringing information to the brain. And then we'll talk later about efferents that take information from the brain to the body. So we're talking about afferents now. And these afferents are all mixed up in a sensory nerve, or in a set of sensory nerves. Say in your arm it would be the median nerve and the ulnar nerve and the radial nerve. They're all a mixture of these the sensory neurons in those nerves are a mixture of pain sensing neurons and temperature sensing neurons and touch, pressure, and position sensing neurons. All the axons are all mixed up, but they all have their cell body in the dorsal root ganglion, and they all enter the dorsal horn. They all enter the spinal cord at the dorsal horn. And there they start to split. now the pain and temperature sensing neurons form a synapse, whereas the touch, pressure, and position sensing neurons don't. The pain sensing neurons, the synapse, the secondary neuron, crosses over, the axon from the secondary neuron crosses over the gray matter and then enters the anterolateral column to the brain. The touch, pressure, and position neurons don't synapse. They enter this gray matter, but then they immediately, their axons go up white matter in the dorsal column. In the pain temperature, the crossing over has happened. The axons of the secondary neuron go up the spinal cord on the contralateral side, contralateral to where the input came from, contralateral side, form a synapse in the contralateral thalamus, processing going on in the thalamus, and then the third order neuron, there's lots of synaptic processing here in the thalamus, and then a third or nth order neuron then goes to the somatosensory cortex in the post-central gyrus of the parietal lobe. In the case of touch, pressure, and position, the primary afferent goes up the ipsilateral side of the spinal cord, forms synapses in the brainstem. The crossing over then happens in the brainstem, processing in the contralateral thalamus, projection of this higher order neuron into the somatosensory cortex, where the information is processed. And so the somatosensory cortex is a mixture, again, of pain, temperature, touch, pressure, and position processing. Each sensory afferent enters the spinal, not each, but sensory afferents enter the spinal cord at different levels, which give rise to this dermatome idea. So, for example, sensory afferents from the neck and shoulder enter the spinal cord at C2, C3, and C4. So those neurons have their dorsal root ganglion here, here, and here, and their sensory afferents enter in this area between these vertebrae at C2, C3, and C4. Right? So that's where those neurons enter. And sensory afferents from the hands and arm, blue area, hands and arm, enter hands and arm, so I'm not talking about shoulder here, so let me do this again. Neck and shoulder, C2, C3, C4. Hands and arm are from C5 to T2. So those afferents are entering in this region of the spinal cord. And then they're going up to the brain, either having crossed over if they're pain temperature, or not if they're touch, pressure, position. Sensory afferents from the torso, T2 through T12. Sensory afferents from the legs and hip, L1 through L5. So here we're entering in this part of the spinal cord and then going up to the brain. And sensory afferents from the heel and the genitals enter at the sacral levels. So sacral 1, sacral 2, 3, 4. Okay, so different sensory afferents from the different parts of the body enter the spinal cord at different levels of the spinal cord. And then the neurons, either the primary or the secondary afferent, depending, go up the spinal cord, either in the dorsal column or in the anterolateral column, depending. Yes?
Does this mean that sacral has fewer axons than cervical?
Yes, it does mean that. So the white matter of the spinal cord looks different in the lumbar. Looks different. The white matter in the spinal cord, in the say lumbar region, which is only carrying white matter from the legs and hip, looks different than the white matter in the cervical region, which is carrying axons from the entire body. So when I show you a figure like this, it's a generic. spinocerebellar tract. So proprioception, our ability to know where our body is in space, is complicated. Some proprioception is carried in the dorsal column, along with touch and pressure information, to the thalamus and to the cortex. But some proprioception is carried to the cerebellum, and these are the spinocerebellar tracts. And they're complicated, so let's just walk through them. There is an anterior, a ventral, an anterior spinocerebellar tract, and a posterior spinocerebellar tract. And in this cartoon, the anterior is cartooned on the right side, and the posterior is cartooned on the left side. But of course, there is a right posterior and a left anterior as well, right? So it's not that they're segregated. The anterior is on both sides, and the posterior spinocerebellar tract is on both sides. In the anterior spinocerebellar tract, the first synapse is in the dorsal horn, gray matter, just like in the pain and temperature sensing axons. The secondary axons cross over they cross over in the spinal cord, and so they go up the contralateral side of the spinal cord. So here is the information. This is somatosensory information from muscles entering, there's the dorsal root ganglion, entering the spinal cord in the dorsal horn, crossing over, going up the anterior spinocerebellar tract. But now something weird happens. In the pons, it crosses over again and ends up in the ipsilateral cerebellar hemisphere. So the left cerebellum is processing information from the left side of the body because of the double cross. Crossing over in the spinal cord and then crossing over again in the pons. So this crossing over is the spinal cord and the pons, a double cross. And so the ipsilateral hemisphere of the cerebellum is processing information about proprioception brought to it from the spinocerebellar tract. The anterior spinocerebellar tract. The posterior spinocerebellar tract, again, axons, cell body, enters, and immediately it again forms a synapse. And then the secondary neurons go up the ipsilateral posterior spinocerebellar tract. And they stay ipsilateral. So again, the ipsilateral cerebellum processes the information from the posterior as well as the anterior. But in the anterior, it's crossed over in the spinal cord and then double crosses back over in the brain. So it's complicated. What do I want you to know? I do want you to know that there is such a thing as an anterior and a posterior spinocerebellar tract. I do want you to know that the spinocerebellar tract projects to the cerebellum, not to the cortex. So that proprioception writ large is processed in the cortex because of the dorsal column proprioception pathway, and it's processed in the cerebellum because of the spinocerebellar pathway. I do want you to know that it ends up in the ipsilateral, not the contralateral cerebellum. Even though proprioception is processed in the contralateral, cortex. I don't need you to know that the anterior does the double cross and the posterior does the no cross. Don't need you to know that distinction. Okay? Yes?
So, I guess I'm just a little confused as to like what it is for compared to what we have what is this sort of?
So what kind of proprioception is this doing as opposed to this proprioception here?
So, right, but so.
It's all proprioception.
So it's only proprioception.
All we've been, so the spinocerebellar is proprioception.
Okay.
The dorsal column carries touch, pressure, and proprioception. Spinocerebellar is proprioception only. And it allows us to do remarkable calculations of how to move our body to do what our cortex is saying we want to do. So when a left fielder is running to catch the ball, it's an amazing set of calculations incorporating the visual system and the proprioceptive system to know how do I move my body to get my body to where that fly ball is going to be in 1 second from now, right? So that it's an amazing set of calculations that our body is good at. And the spinocerebellar pathway is an important part of that calculation. Knowing where our body is, comparing it to where we think our body needs to be, and making adjustments in the motor system. So here's a complicated slide, cross-section of the spinal cord. Right now we're focusing on the blue. The red is motor, so we're going to come back to this when we get to the motor system. So you can ignore the red and focus on the blue. Here's the dorsal column. Here's the right dorsal column and the left dorsal column, carrying touch, pressure, and proprioception information. Here is the right anterolateral column, carrying pain and temperature information. And here's the anterior and the posterior, anterior and posterior spinocerebellar tracts, carrying more proprioception. Those are the key things to know. Dorsal, so right, left and right, left and right. Let's just do one side at a time. Right dorsal column, touch, pressure, proprioception. Right posterior spinocerebellar, proprioception. Right anterior spinocerebellar, proprioception. And right anterolateral, pain and temperature. So there's a cartoon that's a blank. And so we can simplify this, that cartoon. So the dorsal column, let's just do it on the right side. The dorsal column is carrying ipsilateral touch, pressure, and proprioception information to the brain, where it will form a synapse in the midbrain, cross over in the midbrain, and then the information will be processed in the contralateral thalamus and the contralateral cortex. But in the spinal cord, it's ipsilateral. We have the anterolateral column on the right side. Of course, there's an equivalent one on the left side. And that's carrying contralateral pain and temperature information because of the crossover when the neurons entered and then formed a synapse, and then the secondary neurons crossed over and went up this column. And we have the posterior and the anterior spinocerebellar tracts. And some of them are carrying ipsilateral information, and some of them are carrying contralateral information, because some of them are crossed and some of them are not. I'm trying to be vague here because I don't care that you know that it's the posterior that is not crossed and the anterior that is double crossed. You don't need to know that. But some of it is ipsilateral information and some of it is contralateral information in this, going from the spine to the cerebellum proprioception. And the rest of the space we're reserving for other things and for the motor system. The anterolateral column has another name, which you'll see in books, called the spinothalamic tract, which is helpful because it tells you it's going from the spine to the thalamus, which is what it's doing. Just like the name spinocerebellar is going from the spine to the cerebellum. So this is a nomenclature that you'll see maybe a few more times. It's always from to. From the spine to the cerebellum. From the spine to the thalamus. You'll need this kind of information to do the problem set. So now I'll remind you that there's a problem set, which you can pick up here, a hard copy. It's available on Moodle. It's due on Sunday night here at CST. Open book, open notes. You can work with your friends, but you got to turn in your own answers. Okay, we've looked at this one already. The point where we're going now, so we're leaving the spinal cord and going into the brain. The processing in the thalamus is complicated, and I'm just going to leave it at that, because what we're going to do is talk about the mapping of this information in the sensory cortex, in the post-central gyrus of the parietal lobe. That's where we're going now. So here is a cartoon of the brain, looked at from above, front, back, left, right. Longitudinal fissure, frontal lobe, parietal lobe, occipital lobe. Things, words you should know. Central sulcus in this cartoon is here. Post-central gyrus is here. So here's a not quite coronal section, sort of an angled coronal section through the post-central gyrus, and then projected this way. So this is a semi-coronal section, an almost coronal section. You know, coronal would be like this, but they've angled it like this to make sure that it's aligned with the post-central gyrus. So lateral, medial, longitudinal fissure, parietal lobe, lateral fissure, temporal lobe, post-central gyrus in this cartoon version, and a map of the body on the post-central gyrus. So the face and the jaw and the mouth are mapped laterally. And the hands and arm are mapped superior. And the legs and toes are mapped medial in contact in the area next to the longitudinal fissure. And the genitalia are mapped most medial and most ventral in that fissure. And so there is a map of the body. You can see the body's not mapped one-to-one because we dedicate a lot more of our cortex to the face, especially the mouth and the jaw and the tongue, because movement here, sensation here is very important for the formation of language. So a lot of real estate in the neocortex given to the somatosensory system related to the face and the mouth and the hands, but not so much to the arms, torso, and legs, because the receptive fields are bigger. And so the amount of real estate, amount of area of neocortex needed to process those larger receptive fields is smaller. So small receptive fields, lots of cortical processing. Large receptive fields, less cortical processing. You'll need this for the problem set too. I don't want you to memorize the map. If I ever ask you to work with a map on a quiz or a problem or a exam, I would give you something like this. Questions? Small receptive fields allow us to detect fine touch, right? We can discriminate between two very close together points on our lips, but we can't discriminate between two very close together points on our toes. So these have small receptive fields, and we've got millions of them, many of them, and so we use a large area of cortex to process that sensory information. So a large area of cortex for processing just this part of our body, whereas over here we have a similar part of the cortex processing practically the whole rest of our body, except for the hands, which are also fine because of our ability to manipulate with our fingers. Sense and manipulate with our fingers. Does that help? Alex?
If we use our feet more, would we have more space?
If we what?
Use our feet more for things?
Yeah,
would we
I
have
suppose.
more space?
Yes. So there are people who, because of birth defects, are born without arms, and they've learned how to write and play the piano and do all kinds of things with their toes and feet. And so this area of their brain is much expanded because this area of the brain isn't doing what it normally does, or at least it does for a wild type person, right? So this part of the brain, in fact, this part of the brain, which is normally maps from the hands and fingers, might actually receive some mapping from the feet and toes. So in a person who is born without arms and has learned how to use their toes and legs for all kinds of the manipulation that the rest of us use our hands and arms to do, their map could be really different. They can have feet and legs mapped here and here. That's cortical plasticity that would arise from that kind of unusual information input. Okay, let's talk about pain modulation for just a minute, because this comes back to a key concept that I introduced and in fact tested you on the exam. So here is a pain neuron. This is a cartoon of a pain neuron, a free nerve ending in the skin expressing one of those receptors, like either a capsaicin receptor or a ATP receptor to detect painful stimulus in the skin. Cell body in the, where's the cell body? Dorsal root ganglion. Entering what part of the spinal cord? It's the name of where this axon enters the spinal cord. The dorsal horn. Where is this synapse? In the dorsal horn. This neuron is the one that crosses over. Right, so this is an anatomically bad cartoon, but it's a physiologically good one. Do I need to do that again? Is that helpful to do that again? So, ipsilateral, this is where the pain starts. Ipsilateral dorsal root ganglion. Ipsilateral dorsal horn. Ipsilateral dorsal horn is where the synapse is. The secondary neuron crosses over. What's the name? Okay, now it crosses over, goes up to the thalamus in the what? What's the name of this, where this white matter, these myelinated axons are in the spinal cord? The anterolateral column or the spinothalamic tract? I would accept both names. Contralateral, spinothalamic, anterolateral column. Thalamus, cortex. The neurotransmitters that are responsible for transmitting pain information are a peptide neurotransmitter called substance P and glutamate, an excitatory neurotransmitter. So this cartoon focuses on substance P, but glutamate should also be there. So these are excitatory neurotransmission. Activation of pain path neurons, activation of the secondary neurons in the pain pathway, carrying pain information to the brain where our brain says, ouch. Well, our brain says, oh, that hurts. There are modulatory pathways for this. So there's a part of the brain called the periaqueductal gray matter, which I'm not going to ask you to know about, in the reticular formation that I'm not going to ask you to know about. These are efferent neurons coming from the brain to the spinal cord. And these neurons release opioids, enkephalins, endorphins in the spinal cord. So we have here a presynaptic, what do you suppose, presynaptic facilitation or presynaptic inhibition of this synapse? Inhibition, right? We're trying to inhibit the pain pathway. Endogenous opioids, your book uses the wrong word. They aren't opiates, they are opioids. Endogenous opioids activate opioid receptors on the presynaptic terminal of the primary afferent. These opioid receptors are coupled to GI. GI inhibits adenylyl cyclase. The beta-gamma subunit of a GI activates K channels. What is activation of a K channel going to do to this presynaptic terminal? Hyperpolarize it. What is inhibition of pKa going to do? Prevent phosphorylation of calcium channels. The beta-gamma subunit also inhibits calcium channels. So the hyperpolarization and the inhibition of calcium channels, less substance P and glutamate release, less reception of pain information, less transmission of pain information to the brain. Endogenous modulation of the pain pathway. Opiate drugs like oxycodone and heroin and morphine do the same thing. But of course they do more than this because they are addictive and habit forming. Not because of here, but because of what they do in the brain. Okay, so I'm using the fundamental concepts of presynaptic inhibition, in this case, axo-axonic inhibition, to describe how endogenous opioid neurotransmitters, enkephalin and endorphins, modulate the release of pain neurotransmitters, glutamate and substance P. Yes? No, the VTA, the ventral tegmental area, so where Kendra's going is trying to get into the reward pathway, which is about addiction, and that is not in the periaqueductal brain. It's elsewhere in the brain. And when we get to it, I'm not sure, when we get to dopamine more, I might do this again with the reward pathway. Okay, let's do some clickers. Wake up and do some clickers. Okay, I'll give you a hint that I think will help almost everybody in the room. There are 3 correct answers. And now you're down. Interesting. That just one bit of information went from like maybe 2% getting it right to lots more than 2. Yes. A, B, and C are all true statements. Lateral inhibition is stronger. I'm looking at C. Lateral inhibition is stronger in the touch vibration sense than it is in the pain sense. You know from your own personal experience that your ability to detect touch pressure, to discriminate fine information about touch, your receptive fields for touch and pressure are small. Your receptive fields for pain, if you get a cut on your hand and it's inflamed, your whole hand hurts, not just the small area or if you get a splinter, where, one tiny spot of inflammation, but a large area hurts not just because of the spread of inflammatory cytokines, which is also true, but because of the lower level of lateral inhibition in the pain pathway compared to the touch pressure pathway. Each part of the skin is not innervated by the same number because we have small receptive fields and larger receptive fields. The skin of the back is innervated by much fewer sensory neurons than the skin of my lips or fingertips. Okay, any questions about why A, B, and C are true and D is not? Yeah.
So, this might be like a strange question, but what exactly is somatosensory information? I know we've been doing that, but
Yeah.
so how is that only detected by long receptor cells? I'm not sure.
That's the way we're wired. That's the way we developed as embryos and as fetuses and as infants.
So that smell and that sort of stuff is not considered somatosensory?
Smell is not somatosensory. Somatosensory is body sensation. Soma, body, just like cell body. Soma, somato, plural of soma, sensory, sensing our body. Olfactory also has long receptor cells, but is not somatosensory, that's olfactory. Visual does not have long receptor cells, and it's not somatosensory, it's visual. If the question had read, all long receptor cells are somatosensory, carry somatosensory afferents, that would have been a false statement. Okay, in the spinal cord, which of the following is true? Let's do this one. Okay, I'll give you another hint. 3 correct answers. Remember, I won't do this on a quiz or an exam. Axons and sensory neurons enter the spinal cord at this dorsal horn. We talked about that. Specific regions of white matter carry, yeah, so the anterolateral carries pain, temperature, the dorsal column carries touch pressure, position, the spinocerebellar carries position. Those are different sensory modalities carried by different white matter tracts in the spinal cord. Sensory information from the body enters the spinal cord at different levels, yeah. From the arm, it enters at the cervical, thoracic, at the legs, it enters at the lumbar. That's a true statement. All nerve cell axons are in white matter? No, because some of those axons are crossing over in the gray matter. Gray matter is mostly dendrites and cell bodies, but all big red flags should go up whenever you see all. Some of that gray matter are axons that are crossing over. Okay, let's do another one. In the somatosensory system, which of the following are true? A hint, one true statement, but read them carefully. A is not true. The cell bodies of peripheral sensory neurons carrying information to the spinal cord are in the dorsal root ganglion, not the dorsal horn. That's why you have to read that one carefully. Don't stop reading at dorsal. Oh yeah, that's true. The first synapse in the pain pathway is in the dorsal horn gray matter. That's the true statement. All touch pressure, pain, temperature, and proprioception information enters the spinal cord or lumbar region? No, that's not right. Some of it enters at the cervical or thoracic. Touch pressure information is carried in the ipsilateral dorsal column, not the ipsilateral anterolateral. Yes?
I have a question regarding the previous question. So you said that axons of sensory neurons into the spinal cord and dorsal horn.
Yeah.
Is that also the case for the spinocerebellar tract?
Yeah.
Okay.
Okay, and all of these sensory afferents, all of the sensory afferents have their cell bodies in the dorsal root ganglion, and they all enter in the dorsal horn, and then they split up. So pain, temperature form a synapse in the dorsal horn. Touch pressure go up the ipsilateral dorsal column. Proprioception, some of them go up the ipsilateral dorsal column. Some of them cross over into a some of them, no, yeah, some of them form a synapse and cross over, some of them form a synapse and don't cross over, and then they go up the spinocerebellum. Okay, we have a few more, but I think in the interest of time, I'm going to skip them, and you can do them as homework. Oops, that's not enough. So let's take a break now, and we'll transition from somatosensory to visual. So the point is there are additional clickers on PowerPoint, which will be available on Moodle, which you can use to check your knowledge when you're studying this material. So let's take a break, and we'll get to the visual system. From somatosensory cortex and somatosensory system, our ability to convert physical sensation, touch, pressure, pain, position information into electrical signals that then go to the brain and are interpreted by the brain as touch, pressure, pain, or body position. Now we're going to transition to a visual system where our eyes, our retina in particular, convert photons into electrical signals that our brain can interpret. That's where we're going. We'll start with some anatomy, and then we'll get into the cellular and molecular physiology. That's where I like to go, and that's where we will go. But we'll start with anatomy. Here is an inferior view of the brain. Right? So this is an actual brain that's been carefully dissected to show some certain things. What's this? What are these right there? That's olfactory. Right? So we're going to go there, but that's not where we are right now. From the eyeballs are not in the brain, of course. This is my addition. But from the eyes, from the retina, there are optic nerves which have been cut. You've seen it like this in our brain too. The optic nerves were cut. Optic chiasm. There's going to be some interesting things going on here in the optic chiasm. Optic tracts from the optic chiasm to the thalamus. In particular, now we're going to talk about a specific nucleus in the thalamus called the lateral geniculate nucleus, LGN. Lateral geniculate nucleus is a part of the thalamus that is dedicated to processing visual information. So that's an anatomical term that I do want you to know. You know how to find the thalamus in the brain. Maybe I'll show you how to find the lateral geniculate nucleus of the thalamus in the brain, but maybe not, just as long as you know the name associated with the visual system. Is the thalamus associated with the somatosensory system? Yes, but not the lateral geniculate nucleus of the thalamus. The lateral geniculate nucleus of the thalamus is focused, is dedicated to visual system processing. So synapses, lots of synapses. I haven't talked about synapses at all yet, but there's lots of synapses in the retina, synapses in the lateral geniculate nucleus, and then axons from the lateral geniculate nucleus form this thing called the optic radiation to the visual cortex, the primary visual cortex in the occipital lobe. So this is white matter tracts that have been carefully, beautifully dissected to show the connection between the lateral geniculate nucleus of the thalamus to the primary visual cortex in the occipital lobe of the brain. So we'll be using this anatomy over and over again. Now let's focus on the anatomy of the eyeball. Obviously the organ that transduces light photons into electrical signals. So we have to do a little bit of physics. Some of you had this in high school physics, some of you had this in college physics. I'm not going to do a lot of physics, but it has to do with an image. So if you did optics in high school or college physics, I'm going to be using some of the same language, but I'm not going to teach physics. This is a lateral, I guess you'd have to say a sagittal section through an eyeball. Lateral section. This arrow here is supposed to represent an object that this eye is focused on. This object, photons from this object, form an image on the retina, and it's inverted. because that's what lenses do. Those of you who had optics know about this. But let's go through the anatomy. The clear layer on the outside of the eye, the clear layer is called the cornea, and the white layer on the rest of the eye is called the sclera. Sclera, cornea. Cornea is transparent, the sclera is not. Between the cornea and the iris is a muscle that forms the pupil, the dark spot that light goes through. So, you know, the color of your eyes is determined by the properties of the iris. And then the pupil is the hole in the middle of the iris. Hopefully this all is obvious. The chamber between the cornea and the iris is called the anterior chamber, and it's filled with aqueous humor. That's not on here, so you can label you can annotate your handout. This fluid in here is called aqueous humor. Aqueous because it's watery. Humor because that's what some Latin people call it. It's not funny at all. It's just they call it humor. I don't know why. I don't know the root of the Latin word humor. Aqueous humor. Just learn it. So light goes through the cornea. This is not the lens. This is not what does the focusing. This is just the window on the outside of the eye. Aqueous humor. The iris is a muscle that can constrict or dilate. When it constricts, it's a sphincter muscle. So when it constricts, when it contracts, it reduces the diameter of the pupil, limiting the amount of light that comes in. And when it relaxes, it dilates and allows more light in. So in the dark, your brain interprets, oh, it's dark, sends a signal to the iris muscle that we'll talk about in a minute, causing it to relax so that more light can be brought into the retina for better visual processing. When you're out in the bright sunlight, the brain says, oh, that's a lot of light, I don't need that much light, it might actually damage my retina, let's constrict the iris and reduce the amount of light hitting the retina. Okay, but it's a sphincter. Constriction makes it smaller, dilation makes it bigger. The pupil, that is. Behind the iris is the lens. This is what focuses the light this is what focuses the image onto the retina. The curvature of the lens is what allows the lens to focus the image from the object onto the retina. The lens is held to muscle called the ciliary muscle, which is very near, but not and maybe even connected to, but not the same as the iris. So the ciliary muscle is connected to these little tiny little tendons. So they're not really tendons because it's, well, I guess it's tendons. It's connecting muscle to something that's not muscle. Not bone, but it's not muscle. And that puts tension and holds the lens in place behind the iris at the pupil. We'll talk about the focusing of the lens in a moment. The lens focuses the image onto the retina. The contents of this posterior chamber, so anterior chamber, posterior chamber, aqueous humor, vitreous humor. Vitreous because it's not watery. You may know that vitreous means glassy. It's not exactly glassy either. It's more like a gel. And you'll see this in lab either Thursday night or next week, depending on whether you're Thursday or Tuesday. The retina is a complicated set of neurons that actually starts the visual processing. So people say, oh, the eye is like a camera. Well, the eye is not like a camera. This part of it is like a camera, but as soon as we get to the retina, it's not a camera because we're not forming a pixel by pixel image. An image is projected onto the retina, but as soon as we hit the retina, we're starting to convert that image into electrical information. There are synapses in the retina that are processing that visual information. So it's not just a camera. It's a camera plus a image processing system. We'll talk about the layers of the retina in a minute. There is the outside of the retina. There is the retina pigment epithelium is going to be an important nutritive layer for the retina. The choroid is another layer, and then the sclera. So retina, pigment epithelium, choroid, sclera is the layering of the retina at the back of the eyeball. The optic nerve, which is going to go to the optic chiasm. This is white matter axons from the retina projecting to the lateral geniculate nucleus. Okay, so I've used a lot of the anatomical names. Let's look at the eye. So now we're going to focus on how the eyes move to stay focused on an object. We are really good at this. We are really good at putting our eyes where we want them, following a moving object, sweeping across the sky or along the ground. We're good at this. Our eyeballs are really good at tracking. And that's because we have a lot of muscles and a lot of processing dedicated to movement of the eyes. So this is a superior view of the eyes. So anterior, this would be the nose. Posterior, here's the optic chiasm. An idealized superior view of the eyes. There are muscles that control the movement of the eyes. So let's just go through them. So this muscle here, which doesn't have a label on this side, but does have a label on this side, it's the same muscle. It's called the lateral rectus. So this is the right lateral rectus, and this is the left lateral rectus. So you can imagine, contraction of the left lateral rectus would rotate this eye to the left. Contraction of the right lateral rectus would move this eye to the right. We don't do that very much. we don't move our eyes like this. I can't do it even if I tried. So there's systems in our brain that have told me, don't ever contract the left lateral rectus and the right lateral rectus at the same time. If you contract the left lateral rectus to move your eyes to the left, relax the right lateral rectus so that the right eye can move with the left eye in parallel, right? So as you can imagine, there is a medial rectus. Here is the medial rectus of the right eye, and here's a medial rectus of the left eye, which move, so the medial rectus on the right eye moves the right eye to the left, towards the midline. Moves the right eye towards the nose, to the left. And the medial rectus on the left moves the left eye to the right. And obviously I can contract those at the same time and force myself into a cross-eye. That one I can do. I don't like to do it. It doesn't help me see, but that one I've learned how to do. Yeah.
So the lateral rectus is like one of the muscles that is affected with like person with strabismus, right?
Strabismus, both the lateral and the medial rectus can be affected by strabismus.
Okay.
Strabismus is when people, you may have seen people who have trouble maintaining a gaze and their eyes are constantly sort of darting around.
Yeah.
That's strabismus.
Correct.
Their eyes are constantly sort of moving around. That's because they have trouble control of their lateral and medial rectus muscles.
Okay.
So now we've talked about movement of the eyes to the left and to the right. Obviously, there are muscles that move the eyes up and down. So there is the superior rectus. The superior rectus on this eye, when it contracts, it's going to rotate this eyeball upward. So it elevates the eyeball, and there's one over here that's been cut in order to show you deeper muscles on this side. So the superior rectus raises the eyeball. The inferior rectus lowers the eyeball. So we have lateral medial, superior, inferior rectus, moving the eyes. And now we have a couple more that do some complicated things. There's the superior oblique, which helps, it sort of rotates the eyes and sort of causes them to move in. So when we're looking at something very close, not only do we use our medial rectus to sort of go cross-eyed, but we use the other muscles to sort of rotate the eyes a little bit. So lateral, medial, superior, inferior, all very straightforward in a three-dimensional world. The rest of it gets complicated. The superior oblique and the inferior oblique do complicated things related to intorsion and rotation of the eyeballs.
It's usually the inferior oblique is responsible for the downward movement of the eyeball, correct?
The inferior rectus and the inferior oblique are both important for depressed downward movement. Okay, so I will ask you to know the names of these muscles, but they're pretty straightforward. Any questions about this musculature?
Did you say anyone that wasn't superior?
Inferior oblique. It's right, so it's shown here. It's underneath the eyeball. This is the inferior oblique. So here's the superior oblique going through this sort of pulley thing and attaching to the eyeball on the top. And here's the inferior oblique doing the same sort of thing on the bottom. Let's talk about innervation of both the muscles and the iris and the ciliary muscle. So we're going to talk about movement, the nerves required to move the eye, and to focus the eyes, and to constrict the pupil. The three nerves that you need to know are all cranial nerves, so you've learned their names already. The oculomotor nerve, the trochlear nerve, and not shown on here, so annotate. Did I give you this one? No. Right somewhere. The abducens nerve. Okay? Let's start with a simple one. The trochlear nerve innervates the superior oblique, period. The trochlear nerve innervates the superior oblique muscle. Okay, let's do the next simplest one. The abducens nerve innervates the lateral rectus muscles. The name helps you. Abducens, abduction. To abduct the eyes is to move them away from the midline. So the abducens nerve is what causes contraction of the lateral rectus muscles on both sides. That would abduct the eye on both sides, which is a movement I can't do. Bottom line, the abducens nerve innervates the lateral rectus muscles. Everything else in the motor control of the eyes is the oculomotor nerve. So the oculomotor nerve innervates the superior rectus, inferior rectus, medial rectus, inferior oblique. It also innervates a ganglion that innervates the ciliary muscle and the iris muscle. Let me say that again. The oculomotor nerve innervates muscle, like the superior oblique, innervates a ganglion that innervates whose neurons. Remember, a ganglion is a cluster of neurons. The oculomotor nerve innervates a ganglion. It's called the ciliary ganglion. That's not a name I'll ask you to learn. A ganglion that controls the ciliary muscle and the iris muscle. What is the neurotransmitter released by the oculomotor nerve on the superior rectus? Say again? Acetylcholine. It's a muscle. This is a motor neuron. It releases acetylcholine. What type of receptor is on the superior rectus? Nicotinic receptor, just like every other muscle. Superior rectus is here. Here's a muscle that I'm not going to ask you to know, but the levator palpebrae superior is superior. Anybody have an idea what the levator palpebrae superior is? It's the eyelid. It's the upper eyelid. Okay, so we can open up, raise our eyes, open up. Anyway, here is another nerve from the oculomotor nerve innervating this ganglion. What neurotransmitter is released here? Acetylcholine. What type of receptor is here? Nicotinic. This should start to sound familiar. What type of what neurotransmitter is released here? Acetylcholine. What are the receptors here? Muscarinic. This is like the parasympathetic nervous system. Okay? I won't ask you to memorize the neurotransmitters here. I will not ask you to memorize the neurotransmitters, but you might have to use it in a problem, where I would give you this information, or you could look it up and use it in a problem. This is a complicated image, that is everybody, and there are things that we don't need to know. For example, we're not focusing on the trigeminal ganglion. We're not focusing on the sympathetic plexus. We're not focusing on the internal carotid. So there's stuff here that you don't have to know, but there is some things that you do. And I think I've narrated the things that I want you to know. Abducens nerve innervates the lateral rectus. Trochlear nerve innervates the superior oblique. Oculomotor nerve innervates just about everything else. Let's talk about how you focus light onto your retina. How you focus an image onto the retina. So, you know that if you're focusing at a distance, things in the near field of view are out of focus. So, for example, I'm looking in the far corner of the room. I'm looking at Maddie over there. Hi, Maddie. I'm focusing the image of Maddie onto my retina. But I'm also putting my finger in my field of view, which is not on the other side of the room. My finger is right here, which is out of focus, right? Or what I can do is I can focus on my finger, and now Maddie is out of focus, right? So I've changed the shape of my lens in order to focus the image either of Maddie or of my finger. You can do it too. You put your finger here, and then focus on something behind your finger, and then focus on your finger. You are using your motor system to change the shape of the lens. So let's look at a distant object. Here's Maddie's image focused on my retina. the lens is relatively flat. A flat lens allows me to focus distant objects, images from distant objects, onto my retina. To get a flat lens, I have to put, so the lens is suspended by those ligaments, the zonular fibers. To get a flat lens, I have to put a lot of tension on those ligaments in order to pull it flat. It, by itself, it would round up. That's its natural shape, is to be round. But if I put a lot of tension on it from those ligaments, a lot of tension, I can flatten it. So putting a lot of tension on it means dilating the ciliary muscle, right? I want the ciliary muscle to be dilated in order to pull hard on the zonular fibers in order to flatten the lens. Now it gets weird. In order to dilate the ciliary muscle, I have to relax it, right? This is constriction of the ciliary muscle, and this is dilation of the ciliary muscle, just like the iris. Constriction of the ciliary muscle, dilation of the ciliary muscle. Dilation of the ciliary muscle pulls on the fibers and flattens the lens. Constriction of the ciliary muscle relaxes the zonular fibers and allows the lens to round up, which is what we do when we focus on near objects. So what I want you to know is focusing on a near object requires a round lens, which requires a constricted ciliary muscle. Focusing on a distant object requires a flat lens, which requires a relaxed ciliary muscle. And you'll use this in the problem set, because we'll talk about, you'll use this in the problem set. And so obviously, if the lens is not in the right shape, so here's a flat lens not focusing on a nearby object. So you'll use this kind of information for the next problem set due on Sunday. The pupil and the ciliary muscle are different. They do different things, but they're innervated kind of together. So it is very hard, it's difficult for us. We can do it, but it's difficult for us to constrict the pupil and dilate the ciliary muscle at the same time. So remember that I mean, obviously we do it because we can certainly focus on distant objects out in bright sunlight. We can do that. But lots of things, if we mess that up with a drug, and this is getting to the problem set, those of you who have ever been to an optometrist and have had your pupils dilated so that the doctor can look at your retina, how many of you have ever had your pupils dilated? Right, so you've had the experience of not only are your pupils dilated, and then the doctor gives you sunglasses to make sure you don't damage your retina when you go out into the bright sunlight after the appointment, but you can't read anymore because you can't focus on near objects. And I'm going to ask you to work with that on the problem set. So there's a whole bunch of clickers, a whole new set of clickers associated with this part of the visual system that I'm not going to do, because you can do this as homework. Thinking about what muscles move, what nerves fire, because on Thursday, I'm going to want to go to the part of this lecture that I didn't get to, and that is the retina. So for example, we're going to start on Thursday with slide 34. I'm not surprised that I didn't get to it today. There's a lot of material, but we're not going to do the boatload of the 10 slides worth of clickers between slide 24 and slide 34. We'll do that as well. Okay, So some of you I'll see in lab this afternoon, and the rest of you I'll see you on Thursday.